I am very honored to be this year's recipient of the Joe and Betty Ward Award for Excellence in the Study of the Biology of Aging. And it means a lot to me that my research was recognized this year in particular, as earlier this year, my family lost two wonderful women to Alzheimer's disease. So thank you for this honor. My work focuses on the role of the brain vasculature in contributing to the onset and progression of Alzheimer's disease. But this vascular dysfunction is actually occurring prior to the onset of cognitive impairments. So this means that it occurs much earlier than a clinical diagnosis of Alzheimer's disease would be made or even suspected. Alzheimer's disease is the only disease among the top 10 causes of death in the United States with no disease-modifying treatment. And with the exception of one drug, which is Namenda, there is not even a symptom-modifying treatment available. So this leaves the 1 in 9 people over the age of 65 who have Alzheimer's disease with no treatment option. Alzheimer's disease is a chronic progressive neurodegenerative disease, and the pathophysiology includes neurodegeneration, shown here in this picture as brain atrophy, as well as the accumulation of amyloid beta and tau. But what makes things difficult for both treatment and research is that 98% of Alzheimer's disease cases have no known cause. But what our lab and others are starting to uncover is that brain vascular dysfunction plays a large role in both the early onset and progression of Alzheimer's disease. So to highlight the importance of cerebrovascular dysfunction, this is data from the Alzheimer's Disease Neuroimaging Initiative, where they plotted the various biomarker abnormality index across the transition from healthy control to early and late mild cognitive impairment, all the way through a diagnosis of Alzheimer's disease. And over here I have the same data just shown in a different way and at a higher resolution. But what you can see from this study is that vascular dysfunction emerges early on and with the highest level of abnormality compared to the other biomarkers tested. And what's especially remarkable about this study is that there were nearly 1,200 participants and they were followed for over a span of 30 years. So it's a very impressive size study. But these data suggest that vascular dysfunction might be the earliest observable biomarker for Alzheimer's disease, which makes it a potential target for early detection and intervention, as well as a plausible treatment option through interventions that would modify cerebrovascular function. Within Alzheimer's disease, the prominent cerebrovascular changes include decreased resting cerebral blood flow, blood-brain barrier breakdown, diminished cerebrovascular reactivity, and impaired neurovascular coupling. And my talk will focus mainly on the last three. Previous work from our lab has identified mTOR as a major regulator of cerebrovascular damage and dysfunction in Alzheimer's disease. And this figure shows resting cerebral blood flow, and you can see in Alzheimer's disease there's a decrease in resting cerebral blood flow, which is restored with Rapamycin treatment in the J20 mouse model of Alzheimer's disease. So this was our first piece of evidence that mTOR regulates cerebrovascular dysfunction. But resting cerebral blood flow is only one aspect of vascular dysfunction. So my work has been focused on defining the role of mTOR in the other types of vascular dysfunction. So we'll start with blood-brain barrier integrity. But before we get to that, I want to make you aware that we have utilized two mouse models in these studies, the J20 mouse model of Alzheimer's disease and the TG2576. And I've put up some differences between the two models, as well as some of the amyloid beta lesions that are characteristic in the models. But despite the differences, we find similar effects in both models, and that is that mTOR inhibition reduces amyloid beta burden. And so I think having two models of Alzheimer's disease strengthens our studies because it shows our effects are not strain dependent and that they generalize across the mouse models that we have tested. So my first project in the lab was to assess blood-brain barrier dysfunction in 18-month-old TG2576 mice. And what you can see when looking at these two images from the 5-minute time point to the 30-minute time point is that there's no extravasation of the dextran dye between 5 minutes and 30 minutes in our wild type group. But in the Alzheimer's disease mice, there is a dramatic increase in dextran extravasation, indicating significant blood-brain barrier breakdown in this transgenic mouse model, which starts almost immediately after the dye is placed. As you can see, there's high background in the 5-minute time point as well. But blood-brain barrier integrity is completely restored by chronic mTOR inhibition in these mice. We've shown the same thing in the J20 mouse model of Alzheimer's disease as well. So comparing across the 2 time points from 5 minutes and 30 minutes, there's no increase in the wild type group, and we see significant blood-brain barrier breakdown in the J20 mouse model, which is reversed with chronic mTOR inhibition using rapamycin. So this indicates that blood-brain barrier breakdown is driven by mTOR activity in Alzheimer's disease. But we also have strong evidence for mTOR-dependent blood-brain barrier breakdown in other mouse models as well, in diseases with vascular origin. In other words, mTOR-dependent regulation of blood-brain barrier is not specific to Alzheimer's disease. So here we have the LDL receptor knockout mouse model of atherosclerosis, which is a disease originating from peripheral vascular impairment or cardiovascular impairment. And in these LDL receptor knockout mice treated with high-fat diet to induce atherosclerosis, you can see increased fibrinogen immunoreactivity in red outside of the vasculature, which is in green. And that's indicative of blood-brain barrier disruption. And again, blood-brain barrier breakdown is mitigated by mTOR inhibition with rapamycin. So the fact that we find mTOR-dependent blood-brain barrier breakdown in a disease model of cardiovascular dysfunction suggests that mTOR could be a target for intervention in several disorders that share vascular dysfunction as a common etiology. But one of the consequences of blood-brain barrier breakdown in our Alzheimer's disease mice is an increased incidence of microbleeds, and we've shown that in the J20 mouse model as well as the TG2576 mouse model. And we find that in both models, restoration of blood-brain barrier integrity with mTOR inhibition using rapamycin decreases the frequency of microbleeds in the Alzheimer's disease mouse models. So we then investigated the mechanism by which mTOR drives blood-brain barrier breakdown by looking at the expression of tight junction proteins. So rather than there being one physical barrier that restricts entry into the brain through a single access point, the blood-brain barrier is actually maintained throughout the entirety of the vasculature at the level of the vascular endothelium. And tight junctions are located between these endothelial cells, and they're made up of proteins such as occludin, JAMA, claudin, and ZO1. And they help to regulate blood-brain barrier integrity. So our initial studies revealed that the junction's adherens molecule, JAMA, was nearly sensitive to mTOR regulation in J20 Alzheimer's disease mice. We also found that mTOR inhibition with rapamycin increased JAMA expression in the LDL receptor knockout model of atherosclerosis, and we confirmed this mechanism in cell culture, whereby rapamycin, to inhibit mTOR activity, increases JAMA protein expression in endothelial cells. So these studies provide in vivo and in vitro evidence that mTOR activity regulates JAMA expression. But more recently, we've looked at the expression of other tight junction proteins, and much of this work has been performed by Steven Hernandez, who is an undergraduate student in our lab supported by the startup program, and he's sitting right back here. But what he has found is that mTOR attenuation with rapamycin increases the expression of the tight junction proteins ZO1, occludin, and claudin-5, specifically in the Alzheimer's disease mice. So to go deeper into this mechanism, we looked at MMP9 activity, which is well known to disrupt blood-brain barrier integrity through tight junction protein modification. We used an in vivo model of the blood-brain barrier and determined the role of amyloid beta at different levels of mTOR activity. So we used rapamycin to inhibit mTOR and leucine in excess of what's contained in the media to activate mTOR. And we found that in control cells, amyloid beta disrupted blood-brain barrier permeability, and rapamycin treatment prevents A-beta-induced barrier disruption. We also found that overactivation of mTOR with leucine was sufficient to disrupt blood-brain barrier integrity, and also in the presence of amyloid beta. But when we inhibit MMP9 activity, we find that the barrier disruption that was induced by amyloid beta in control cells or overactivation of mTOR or their combination has been returned to control levels. So to summarize this section before we move on, these studies have shown that mTOR drives blood-brain barrier breakdown through activation of MMP9, which alters tight junction protein expression and leads to blood-brain barrier breakdown. So for this last segment, I'm going to focus on the role of nitric oxide synthase in mediating different types of vasodilation. I'll focus on two different types. The first of which is endothelial nitric oxide synthase, which is responsible for vascular reactivity, as well as neuronal nitric oxide synthase, which has an important role in neurovascular coupling, along with eNOS as well. So we'll start with the role of eNOS. These data are from the TG2576 mice we previously saw, but in order to assess vascular function in this case, we applied acetylcholine to elicit endothelium-dependent vasodilation through eNOS activation. And first off, you can see in the wild-type animals that chronic mTOR inhibition with rapamycin leads to increased vasoreactivity in response to acetylcholine. But in the transgenic group, the first thing to point out is that in the absence of amyloid beta, they are performing as well as the wild-type animals of the same treatment condition. So it really is the presence of fibrillar amyloid beta on the vasculature that is inhibiting endothelium-dependent vasodilation. But what really stands out is that attenuation of mTOR with rapamycin mitigates the impairment produced by this fibrillar amyloid beta. So in the control-treated mice, you see that A-beta inhibits vasodilation by 75%, but it's only about a 25% reduction in the mice treated with rapamycin. So here we see that A-beta is directly impairing endothelium-dependent vasodilation, and this impairment is driven by mTOR activity. But how does rapamycin preserve vascular reactivity? So we've shown that rapamycin might help to maintain endothelium-dependent vascular reactivity through direct activation of eNOS. So in this first study, we showed that rapamycin elicits anywhere from a 10 to 30% increase in vessel diameter in vivo. And rapamycin stimulates nitric oxide production in the vasculature, which is indicated by DAF-FM in green, and these areas that have NO reactivity subsequently vasodilate. And in cell culture, we confirmed that rapamycin phosphorylates eNOS in endothelial cells. So this suggests that inhibition of mTOR may preserve vascular reactivity through eNOS activation. And in fact, the restoration of nitric oxide synthase by rapamycin is critical for the rescue in cerebral blood flow in Alzheimer's disease mice. So this first two pictures over here I showed previously at the beginning of my talk, and the similar data is shown on the right. But here we have, with resting cerebral blood flow decreased in the Alzheimer's disease mice, it's restored with mTOR inhibition with rapamycin, but if we block nitric oxide synthase in an Alzheimer's disease mouse treated with rapamycin, cerebral blood flow is not restored back to control levels. So mTOR is altering nitric oxide synthase activity in Alzheimer's disease to produce impairments in cerebral blood flow. And an important form of regulated cerebral blood flow is neurovascular coupling, which is the local increase in cerebral blood flow due to neuronal activity. And this is also mediated by nitric oxide synthase, but in this case, nNOS plays a larger role, and that's neuronal nitric oxide synthase. And eNOS plays a small role as well. So in this first study, I tested whether mTOR was involved in neurovascular coupling impairments in 12-month-old mice that received 8 months of rapamycin treatment. So here you can see the change in cortical cerebral blood flow in response to whisker stimulation, shown in red, in a wild-type mouse. And we find that in the J20 model of Alzheimer's disease, neurovascular coupling is significantly reduced. and neurovascular coupling is restored and even enhanced in J20 mice that received rapamycin. But like I mentioned before, we were concerned about the role of nitric oxide synthase in neurovascular coupling. So we used NPA to inhibit eNOS, and we calculated the eNOS-dependent portion through the application of MPA plus LNAME. So looking at the quantification, you can see that in the wild-type animals, the inhibitors inhibit neurovascular coupling, which is what you would expect. However, in the Alzheimer's disease mice, they are insensitive to the inhibition produced by nitric oxide synthase inhibitors, which indicates a dysfunction in nitric oxide synthase activity. And you can see that mTOR inhibition for 8 months restores nitric oxide synthase activity. So to show this effect more clearly, I calculated the percent contribution to neurovascular coupling, and here you can see very simply that there is a reduction in the eNOS-dependent component of neurovascular coupling in Alzheimer's disease, and that's restored with chronic mTOR inhibition with rapamycin. But I then wanted to test whether mTOR inhibition could reverse neurovascular coupling deficits that were already present in these J20 mice. So these mice are the same age as previous, they're 12 months old, but I only gave them 8 weeks of rapamycin treatment. And you can see a similar effect. Wild type is up here. There's a decrease with Alzheimer's disease, and mTOR inhibition improves and even enhances this effect. And if we look at the role of eNOS and eNOS, we see the same pattern emerge where there's a deficit in neuronal nitric oxide synthase mediated neurovascular coupling, which is restored in rapamycin. And interestingly, in this age group, in the 12-month-old mice with the 8-week rapamycin treatment, there's even an enhancement of eNOS activity in neurovascular coupling in these animals. But now, I also assessed cognitive impairments in these mice, and not surprisingly, at 12 months old, these animals do have memory impairments that are restored by rapamycin treatment. But now I wanted to put our central theory to the test. I wanted to test our hypothesis that cerebrovascular impairments are occurring very early on and prior to cognitive impairments. So for this last portion, I used the same 8-week treatment, but these mice are only 6 months old. And I have a wild type plotted up here. There's a decrease with Alzheimer's disease and a restoration with mTOR inhibition. And looking at the role of nitric oxide, the same pattern emerges even at 6 months of age. So there's a deficit in neuronal nitric oxide synthase in the Alzheimer's disease mice, which is restored and even enhanced at 6 months in this particular study in the mice treated with rapamycin. And importantly, these effects are occurring prior to the onset of cognitive impairment. So 6-month-old J20 mice do not have memory deficits, but they are showing neurovascular coupling impairments. So this really does support a vascular-mediated theory of Alzheimer's disease, where cerebrovascular dysfunction emerges early on in the transition from non-pathological aging to mild cognitive impairment and even Alzheimer's disease. So to summarize this last portion, we have evidence that mTOR inhibits eNOS activation, which leads to deficits in neurovascular coupling that can impact cognitive function and neuronal health. mTOR also inhibits eNOS activation, leading to reduced cerebral blood flow and decreased vascular reactivity, which can impact amyloid beta clearance, and that feeds back to further worsen cerebral blood flow. And we've also shown that eNOS can affect neurovascular coupling as well. So with that, I would like to acknowledge the members of the Galvin lab, Jim Lechleiter and his lab, the optical imaging facility, which is run by Dr. Jim Lechleiter, and I'd like to thank Exing Wong for his technical help, technical expertise. And the H&E staining that I showed was performed by the Nathan Schock Center Pathology Core, directed by Dr. Aquino, and Colton did a wonderful job on those slides. I'd also like to acknowledge all of our funding sources, especially the Biology of Aging Training Grant and my new funding source, the Alzheimer's Association Research Fellowship. So with that, thank you, and I will take any questions.